Hello friends, welcome to my channel Clinical Biochemistry by Dr. P.K. Prabhakar. Today I am going to start a new series of lectures on genetic engineering. So this will have a number of lectures from basics, then we will talk about all the tools and techniques which we are going to use for gene cloning or genetic engineering one by one, and then later on we will talk about the details, the molecular biology and molecular details of that one. So this is the first lecture. Okay, now if you have not subscribed my channel, you can subscribe it so that you will get, and you can press the bell button so that you will get all the notifications as soon as I will upload the video. So first, today just briefly we are going to see what is gene cloning. As we know, cloning means the making the copy, and when we are talking about gene cloning, it is molecular xeroxing, you can call it. So the production of the exact copy of a particular gene is called as gene cloning, or the term, there are number of terms which is going to be used together, like gene cloning. Sometimes we are using DNA cloning, sometimes we are using molecular cloning, recombinant DNA technology, RDT. All these are refers to the same technique, same process. Gene cloning, DNA cloning, molecular cloning, RDT, recombinant DNA technology, everything is same. Okay. So there are a few points. Gene cloning or molecular cloning is a techniques used to make identical or similar copy of DNA or gene. So you are going to make the exact copy, like xeroxing we are going to call it, and it is exactly a molecular cloning, molecular xeroxing you can call it. A fragment of DNA containing the gene to be cloned, that normally we are going to call in molecular biology or gene cloning, we are calling it gene of interest. So the fragment of DNA which contains a gene to be cloned is inserted on a vehicle, that is called as vector, and ultimately the gene of interest plus vector, या gene of interest loaded on the vector is called as recombinant DNA molecule or recombinant DNA. Then the vector is transported the gene into the host cells where you want to clone it or where you want to express it, which is usually a bacterium, although other types of living cells can be also be used, like fungus also we are going to use, Saccharomyces cerevisiae we are going to use, Pichia pastoris we are using it, number of other yeast cells we are using, higher organism also we are going to use it, but most preferably it is a bacterial cells because its replication time is very less. Second, its genetic size, genome size is very small, so that it is very easy to handle it, easy to manipulate it. That's why bacterium is preferred over the other host cells. Then once this recombinant DNA will be transported into the bacterial cells or the host cells, there the host cells, the vector multiplies, producing the numbers of identical copies, not only of its own but also the gene which it is going to carry, that is the gene of interest. So multiple copy of gene of interest we are going to get through this process. When the host cell divides, copy of the recombinant DNA molecules are passed to the progeny and further vector replication takes place. After a large number of cell division, a colony or a clone or identical host cell is produced. Each cell in the clone contains one or more copy of the recombinant DNA molecule. If you see there are DNA cloning. So when DNA is extracted from an organism, all the genes are obtained. From that all the genes, the gene cloning we requires a particular gene of interest. Okay. So the total steps, if you see in the gene cloning, there are 7 steps we are having, preferably. Means if you divide all the steps one by one, so total 7 steps we are having. DNA cloning can be achieved by 2 different methods, that is cell based DNA cloning, or second one is PCR. So here we are going to talk about cell based technique, cell based DNA cloning. I am not talking about the PCR. Okay, so in this case 4 basic requirements. First requirement is DNA fragment, that is containing the gene of interest. Second one is the tools, different type of tools. The most preferred tools are restriction endonuclease and the ligase enzyme. Later on I will come to what is restriction enzyme and what is ligase. Third, vector, and fourth, host where you are going to transfer it. So 4 things: gene of interest, restriction enzymes or the tools, vector means carrier, and the host where gene has to be cloned. Okay. Now come to the steps of the gene cloning. The 7 basic steps of gene cloning is, the first is isolation of the DNA fragment to be cloned, means that is my gene of interest. So first isolate, cut the gene of interest from the specific place, from where whatever gene you want, either you want eukaryotic cells from genes, or you can want prokaryotic genes, whatever genes you want to be cloned, that need to be isolated. Then second is insertion of the isolated DNA onto a suitable vectors. There are a number of different types of vectors we are having. You cannot use all the vectors at a time. So vectors we are going to choose depending on the length of the DNA which we want to clone, where you want to clone, which type of product we want. Number of factors are there. On the basis of that one, we are going to decide which type of vector we need to use it. So insertion of the isolated DNA onto a suitable vector to form a recombinant DNA molecule. Second step. Third step is introduction of recombinant DNA into the suitable organism known as host. Then you are going to select the transformed cells, means those cells which have the recombinant DNA, and those cells which does not have recombinant DNA, that to be discarded. So selection should be there. Then multiplication, expression of the introduced gene into the host. Sixth is isolation of multiple gene clone copies, protein expression into the genes. And last one is purification of the isolated gene, or if you want protein, then protein. So this is the 7 steps. So one by one we are going, this is sequential steps. If you see in this picture, we are having here the first step is, in this case, this is the, from here I am going to isolate a gene of interest. So I have taken a gene of interest. That gene of interest will be isolated and it will be inserted onto a vector. Here vector is a plasmid which has taken from the bacterial cells. So first gene of interest, second vector has been taken. In the third step, vector is loaded with the gene of interest. Then this vector loaded loaded with the gene of interest, he need to be transported into the bacterial cells. Then this bacterial cells will be selected, those bacterial cells which has this gene of interest, and those cells which does not have this gene of interest need to be discarded. And then we are going to clone it, culture it, and then protein or the genes to be isolated. So either gene of interest will be isolated or protein will be harvested, and that protein that will be used for many different places. This is the basic process of gene molecular cloning. Later on I will make a video on all the 7 steps one by one in the details, so where we will talk about more details about the 7 steps. Now probably this picture everyone of you might be knowing that this is the picture of Dolly, the first cloned sheep. This was the cloning only. So the process was like that one. In this case, this sheep had donated their egg. From this one, nucleus has been removed, so it is enucleated egg cells. Then this is nuclear donor. So from here cell has been taken from the udder cells and mammary cells. From this one nucleus has been taken. So this is nucleus from this cells, and the cytoplasmic and other component except nucleus from this cell. Both cells, both nucleus will be fused with the fluid, and then after the fusion it is going to do the differentiation and proliferation, and ultimately it will be implanted into the surrogate Eve's uterus, and then Dolly came into the picture. Dolly's birth has been taken. So this was the process, this was the story of Dolly. The application of gene cloning, if you see, there are many applications. We are having many different fields, we are having different type of applications. A particular especially in medicine fields or the vaccines, like insulin, number of proteins, many number of enzymes we are going to produce by recombinant DNA technology, genetic engineering. Vaccines, currently you might have heard about the COVID vaccines. Almost all the types of all the different types of COVID vaccines are produced by gene cloning only. Then production of many vitamins, hormones, antibiotics, number of interferons, cytokines, all these are for the treatment of some of the specific disease with the help of gene therapy like leukemia and sickle cell anemia. There gene cloning is required. Then we are going to produce genetically modified organism. It will be transgenic animal as well as transgenic plants where gene cloning is going to be utilized. Then we are having in the agricultural fields especially for the transferring of nitrogen fixation genes from the bacteria to the from the leguminous plant to the different cereals, so that easily nitrogen will be absorbed into the plants and plants will get the nitrogen, and that will help into the improvement of the quality of the or the growth of the farm. Then we are going to use pesticides like Bt cotton is one of the example where bacillus toxin gene has been taken from the Bacillus thuringiensis bacteria and it will be transferred into the cotton plant, so that cotton plant will produce the toxin, and whenever an insect will eat the leaf of the cotton plant, insect will die, so that is called as Bt cotton. Then we are also having herbicide tolerance gene from Salmonella has resistance from the glyphosate. Herbicide resistance refer to the inherited ability of wheat to crop biotype to survive and herbicide application, that is original population was susceptible to. There we are going to use one gene has been transferred from Salmonella to the glyphosate to the plant. Then for the bioremediation we are going to use gene cloning, genome organization and gene expression. We are going to use identification of many human disease on the basis of genes clonings. We are going to use like BRCA, that is breast cancer gene. There also we are going to use this gene cloning. Other than this one, genome sequencing and studying of human genes, there gene cloning has been used. Now one of the example you can see it is golden rice. In this case, golden rice was created by transforming the normal rice with 2 beta carotene biosynthesis gene. One is phytoin synthase from the daffodils, and second one is phytoin desaturase from the soil bacterium. So that ultimately in this rice carotene will be produced, and those who will eat this carotene, they will get vitamin E easily, so it will help in the color night blindness. If you see this picture, this is example of tomato, where on the same plant below the soil you will have potato, and on the top of the plant you will have tomato. So potato plus tomato, it becomes pomato. Here we are having friendly insecticide. Just now I have given you Bacillus thuringiensis example where Bt cotton, the toxin gene from Bacillus thuringiensis is going to be transferred into the cotton plant, and when an insect eats it, insect will die. Then this is the resistance for the herbicide. Just now I have told you glyphosate genes will be transferred from Salmonella to the different plants, especially corns, cotton, soybeans, so that they will be resistant towards the herbicides. Then for bioremediations, many different elements can be utilized, for many different elements, many different kind of minerals, metals we are going to use, and there we are having almost 100% efficacy. Then for insulin productions also we are going to use genetic engineering, gene cloning, where human insulin we are going to produce. So insulin gene will be taken from the pancreatic cells or any other cells, because insulin gene is present in almost all the cells, but it is not expressed, it is expressed only in the pancreatic cells. So we are going to take the insulin gene, and then it will be transferred with the help of plasmid to the bacterial cells, and bacteria is going to express that particular protein, insulin will be produced, and then we are going to purify it. You are going to characterize it and purify it. Then recombinant vaccines we are going to produce. Many number of recombinant vaccines where from the bacteria, from the virus, we are going to take particular gene of interest, and that gene will be transferred to the bacteria. So bacteria will produce the proteins, protein will be purified, and that protein will be used as a vaccine. For then virus based biopharmaceuticals normally we are going to use, where replication defective vectors we are going to use for gene therapy, replication competent vectors we can use for oncogenic virotherapy, vaccine vectors for antigen delivery and display, that is vaccines, virus like particles for antigen display vaccines, than inactivated or attenuated virus vaccines. So this is for COVID, SARS-CoV-2. Many different, currently we are going to use this vaccines inactivated virus, that is Covaxin is inactivated virus. Then virus like particles we are having, live attenuated virus, DNA virus we are having, mRNA virus we are having, protein vaccines we are having. So many different vaccines we have produced with the help of gene cloning. This picture gives you a picture, what we call it, a kind of a Spider-Man. This is called as a spider goat. A spider goat means, normally if you see the spider waves, that is 6 times more stronger than the same thickness of steel. Many different types of spider waves are there. Spider waves will have of different chemical composition and different forms are there. So depending on your requirement, you can choose, you can make it. These spider waves or the material which is used in the spider silk, that silk that is 5 times stronger than the steel, can be used at many places. Sorry, it can be used for aeroplane or many other purposes. How it is going to be produced? This is a spider. So a spider, this silk protein is going to be produced. So we are going to take the protein. So in this case, we are going to take the silk protein gene for the silk protein from the spider, and from this goat A we are going to take the cell. From this cells we are going to just take the nucleus. Cytoplasm will be discarded. Then this silk protein gene will be transferred into this nucleus. So we are having a nucleus, hybrid nucleus, or you can call it recombinant nucleus, where the silk protein gene is there. Now from second goat we are going to take the cells which is enucleated cells. So we are going to remove the nucleus. Only cytoplasmic components we are going to take in, and then this nucleus will be inserted into this one. So we are having a hybrid cells which contains 3 different components from 3 different places. Cytoplasmic portions from this goat, main nucleus from this goat, and the protein gene, spider silk gene from this spider, and then it will be cultured and it will be transferred into the different goats and other places wherever you want. Ultimately, when this goat is going to milking, so milk contains that spider protein or silk protein, and that will be either purified or it will be drinking. Then also we are going to use DNA vaccines, where DNA will be isolated from the particular organism and it will be transferred to the in the form of vector. After cloning it can be used. Not only that one, many medically useful products of biotechnology is going to be produced, like colony stimulating factor, which is going to stimulate for white blood cells in the patient with cancer and AIDS. Erythropoietin, which is normally prevent the anemia because it is going to activate the RBC production. Then factor 8, for blood clotting. Growth hormone, insulin, platelet derived growth factor, tissue plasminogen activators, and many other vaccines. All these are products of gene cloning and biotechnology or RDT only. These are other genetically modified products that will be, because these are the product of again gene cloning and molecular biology or biotechnology only. Now for all these 7 steps, whatever 7 steps you have seen, we require 4 important things. That 4 important things were gene of interest, then tools, then we require a vector, and we require host. So overall these 4 things, especially in the tools, we are going to use many other things only, like this is your gene of interest, DNA molecule, vector, host cells you are having. 3 things already there, and 4th is tools. In the tools, many enzymes we are going to use, like restriction endonuclease. This is one of the most important enzymes which we are going to use for genetic engineering. Then DNA ligase for joining, reverse transcriptase, where we are going to use RNA to DNA conversion, like cDNA conversion, that is going to done by reverse transcriptase. Then we are having alkaline phosphatase and kinase, 2 different enzymes. Both are having opposite function. Phosphatase normally removes phosphate, kinase normally adds phosphate. This alkaline phosphatase removes phosphate from the 5' phosphate end and make it 5 prime OH. So 5 prime phosphate becomes 5 prime OH by the help of alkaline phosphatase. And T4 polynucleotide kinase normally convert this 5 prime OH into again 5 prime phosphate. So these both are having opposite activity. Then we are having S1 nuclease. This is going to remove the protein both 3 prime and 5 prime, so that make blunt ended DNA. Cleaning fragment of E. coli DNA polymerase 1, it makes single stranded protruding ends by double ends by extending the shorter strands. Lambda exonuclease, we are having 3 important exonuclease here. Exonuclease normally removes the DNA from the one of the end, either 5 prime end or the 3 prime end. Lambda exonuclease removes nucleotide from 5 prime end. E. coli exonuclease 3 removes from 3 prime end. Exonuclease BAL3, it makes a DNA fragment blunt end from both the end. TDT, terminal deoxyribonucleotide transferase, addition of single stranded sequence to the 3 prime blunt end. And lastly, we are going to use a small DNA fragment that is called a linker and adapter. Later on, each one enzymes I will make a separate video and then I will upload it. So this was the just basics of the briefly what is gene cloning, where we are going to use it, what are the different type of product we are going to get it. So this is just application of gene cloning. Hope you have understand what is the requirement of gene cloning, where we are going to use it, what are the different tools, techniques is going to be utilized. So this is all about for today. If you have any query, any comments, you can write it in the comment box. If you have not subscribed my channel, you can subscribe it. So thank you very much. Have a nice day.